If I could have your attention really quick for a minute, I want to ask you a favor. So I hope all of you, I know because I've gotten a lot of feedback from you, have gone over to see Lynn Denio's work. And from what I've heard from a lot of you is very impressed by it. And so I just wanted to let you know that she is watching the live stream right now. And so I would like you to give her a round of applause for that beautiful work. So Lynn, that's for you. These folks here love the beautiful work that you've created and given to us to reflect on. So thank you. Now, Sasha is going to take us to the next spot.
Thank you. Hello, everybody. This is the first time I've grabbed the mic without asking a question. So I'd like to introduce myself. I'm Sasha Rabchevsky. I'm a professor of physiology at the University of Kentucky in Lexington, sister school, quote unquote, of Louisville in Kentucky. I'm here to introduce you all to a new program that has been developed here in Minnesota, but with a little background. When I was in Paris, France in '99, when I was doing my first postdoc, and I came back to the United States, I had to decide where I was going to do for first postdoc. And there were lots of opportunities, but one of the places I chose, which is the University of Kentucky, was because they had a state fund that was just developed in '99 when I moved there, which they were taking speeding and drunk driving tickets and tariffs and applying them towards research for spinal cord injury and traumatic brain injury research. And this was the first opportunity I had seen this. This had been developed at the University of Florida when I was a Gator, actually back in the day. And Dick Bungie and Paul Reier had been using those funds. And that continued, and it's continued out to many other states across the country. And so Minnesota is one of the most recent states that has begun this program. And what we're going to offer you now and welcome you to visit right now is a poster session of individuals who have been funded by this Minnesota State Fund through the works of Unite to Fight Paralysis and other advocacy groups to show you the quality of the work that they're doing. And again, this is a very interesting opportunity because when I got to Lexington back in 1999, assistant professor, not even, I was an associate at the time. I had no monies. I couldn't get an NIH fund because I had no state funding. So the University of Kentucky and the University of Louisville had these competitive grant systems, which you could apply for grants. And I did. My very first grant was the K-SHIRT, it was called, Kentucky Spinal Cord Injury Heritage Research Trust. And using that money, I developed then preliminary data to get grants from the ISRT in London and from the NIH and from the DoD. So these are meant to be, at one point in time, very seed grants to be able to allow people to help them start their careers. It's actually become much more than that now because actually even PI, established investigators, are using these funds not to start up their labs, but to be able to do experiments that aren't necessarily ready for prime time, if you will, and that they need to generate more data, and the state funds are used to do this. So without touting any more of either UK or U of L or any other entity, I want you all to take the opportunity to go witness these new investigators that are funded by new resources to be able to give them more support in understanding that this is something that must be done. And I think that this can be done virtually in most states, not every state, but the fact is that we needed a template. Matthew has exemplified this, that you can take this and boom it, even double or triple your investments. And I think this is something that it starts off fledgling. It is it's a lot of work. Obviously, you've seen the grassroots, but once it happens, it spreads fast because there are lots of people who want this information who don't know how to access it. So please take this opportunity in addition to networking to go ahead and applaud these investigators and the project that's been going on. So please welcome, it's all out here. I don't know if there's any beverages out there, but at least go with your beverage. Okay, thank you very much.
Come on back in. Let's get started. That poster session was pretty cool, wasn't it? Yeah, we've never done that before. That was very cool. Yeah. Whose idea was that? Mine. You. We can get them. We'll get them. We'll ask the students. In fact, okay. We're just going to get started. There aren't too many folks back over there. Really quick, couple things I want to remind you of. Our survey, please fill it out. Before our sponsors, if they're not already gone and packed up and left, thank them again. Go visit, say hello. What was the other thing? Oh, reminder, just because I know it's going to require some explanation. I brought this up yesterday. I don't think I brought it up today. You have a color on your ID badge. That color corresponds to where you will be directed to sit when we do our breakouts, break-ins. As I mentioned yesterday, in an effort to not have to make you decide which conversation you'd like to be a part of, we will put, for instance, red over here, yellow, blue, green, and we will every 20 to 25 minutes rotate through with a couple of people staying there to moderate the conversation. Conversation on risk benefit for clinicians and people with SEI, advocacy opportunities, the regeneration panel that we'll have later this afternoon, and something else. I can't remember. I don't think so. Not yet. Almost. Oh, yeah. Yeah. Jason's going to give a talk first. Yeah. You'll come up after. Yeah. Good. Hi. Yeah. You're welcome. But for right now, that will come later. For right now, I'd like to introduce you to Jason Stoffer. Some of you have heard him on the podcast. He's our podcast co-host and also Cure Advocacy Network manager. Take it away, Jason.
Come on, Mike. Wow. Okay. Well, I'll just stay here and you guys can adjust because you're good at that. So most of you know that the theme for this year's symposium is a celebration of advocacy. So it's appropriate to do a quick talk on advocacy and what drives us to do this work, what motivates us, and also maybe some of the things that hold us back. So if we can identify some of those. My name is Jason Stoffer. I work for U2FP. I am a white male, not a lot of hair on the top of my head. I guess I've been doing eye color blue. I'm a wheelchair user, but right now I try to get out of the thing as often as possible, so I'm in a wheelchair, and my wheelchair is beside me. So if you talk to anybody who knows us, it won't take you long to discover that me and my buddy and fellow advocate and U2FP board member Quinn Brett have a shared passion for the great outdoors, for physical challenge, and the depth of relationships that are cultivated when those two things are combined. Next slide. Over the years, we've conveyed that passion into action on multiple adventures. And about 2 weeks ago, we culminated a proper adventure, adaptive hiking the Grand Canyon from the South Rim down to the Colorado and to the North Rim. Quinn and I and our friend Ben used our hand cycle trikes, think outdoor wheelchairs. And we were joined by 5 incredible friends. Next slide. The whole adventure took about 2 days. Our route took 25 miles, a loss of 4,500 vertical feet, a gain of 6,000 on the North Rim. Hundreds of switchbacks, innumerable steps and drop-offs. Go to the next slide. Actually, you can go back to that other one, sorry. The whole adventure took about 2 days. I think I told you this already, 25 miles, lots of elevation loss, lots of elevation gain. The trail was difficult but fulfilling, and as far as I know, we were the only folks that have ever done that on hand cycles. The Grand Canyon experience was a story all on its own, but a few things stood out to me that really illustrate the advocacy work that we're trying to do. Next slide. So the first of the things that jumped out were just all of the comments. Thousands of comments. They were, wow, what are those things? Good for you. Awesome. How are you going to get those things out of here? Did you get a permit for those? Everyone said something. People were incredulous. They were confused. They were inspired. Some were really annoyed. But why the compulsion to make a comment? I think it's because no one has ever seen a contraption like our outdoor wheelchairs on that trail. The trail is not built to accommodate us, despite the fact that we can legally access it. The experience is nowhere near normalized. That's what it comes down to. We were the absolute exception. So paralyzed folks with their accessibility devices in a place as rugged as the Grand Canyon is basically a frontier. So this brought to mind the inaccessibility cycle, of which I'm sure most of you are familiar with, but it has a deep connection to our work in research advocacy. Next slide. Okay, let's go over it quickly. You guys have, I'm sure some of you have seen this before, but it's super illustrative. On the top, let's start at the top. So inaccessibility to a space or an idea or an institution. Disabled people are, if they're unable to participate, obviously then disabled people are not visible in that space or that institution or that thought. Okay, moving over. So if they're not visible there, then disabled people are seen as an outlier or a rarity, right? So if now the disabled people are an outlier or a rarity, well, there's no reason to really consider them, to accommodate for them. And then it's back to inaccessibility. So let's go to the next slide. So if the parallels aren't evident, here it goes. If there are no opportunities to voice lived experience priorities in research dialogues, then our opinions and our voice will be absent. If our voice is absent, it appears that we have nothing of value to contribute, or possibly that we're even content with what's going on. If we appear to be content and have nothing to contribute, then why would opportunities to voice our opinion even be created? So there are no opportunities. Kind of see the parallels between the inaccessibility cycle and our voice in the research dialogue. To the extent that our voice is missing in that dialogue about cure research, those that are directly involved in this space, namely clinicians, researchers, government institutions, funders, biotech, et cetera, then they're all operating in an echo chamber. Decisions regarding our community will be made based on what those stakeholders believe to be appropriate for our community. If you want to deep dive into that, you guys should check out our CureCast episode with Kim Anderson about how bad that was years ago. It's gotten a lot better. So you can have the most well-meaning folks in the world on the job, but if the people with the most to gain or lose in the equation are absent, the product will obviously be lacking. The wrong stuff will get funded. Clinical study designs will be found wanting. Recruitment will be lacking. Mediocre results will be hyped in the news. Entropy will rule the day. Your voice is the energy required in the system to optimize it. Next slide. Okay, so let's explore the lived experience role in this cycle. Why do we disappear? Why do we remain silent? In my own experience, I think the gravitational pull away from cure thought was a well-meaning but crippling clinical institutional message. I was provided with this fancy wheelchair right here. I was given world-class education on activities of daily living, ADLs, and skills for independence, as well as access to new cool hobbies and ways to find contentment. What was oddly but conspicuously lacking was any guidance toward work being done to make me whole again physically. Even with my own community, at the mention of the word cure, I would get a look like, we don't talk about that here. That's not a road that you want to go down, kind of a dead end. Some folks got the opposite, but just as devastating message, which was, don't worry about it, man. You're going to be walking in 5 years. So our elder statesmen have seen the hype news cycle so much that it surprised me to see anyone with more than 15 years with this injury still doing this work. Thank you for sticking with it, you guys. For some, just the overwhelming burden of an SCI sucks up all the oxygen in the room. And there's no margin left at the end of the day. So we remain in a slumber. Next slide. So whatever the reasons our community is silent, whatever paved path to apathy got us here, it doesn't mean that we should stay that way or that we shouldn't have higher expectations of the stakeholders. It is our responsibility to wake up and get involved. It's not our job to be pessimists or optimists, but realists. It's our job to voice the simple truth of what it's like to live with this thing day in and day out. It's not an immediate death for most, but an everyday slow one. It's the excruciating pain, and it's the embarrassment and inconvenience that comes with loss of bowel and bladder control. It's the major decrease in lifespan. It's having no physical or mental margin at the end of the day. It's wondering whether your caretaker is going to show up that day and being completely at their mercy when they do. It's every secondary condition. It's the daily assessment and question for some whether to keep going. The actual truth is untenable and it's unacceptable without any added flourish. So what am I getting at with all of this? I'm saying that our voice is not only helpful, but it is imperative for changing the landscape of this injury, for our generation of wheelies, but also the ones that are coming behind us, because they're coming. Next slide. Another thing that occurred to me about the Grand Canyon adventure is this idea of risk versus reward. So how do you find the motivation to say yes to, say, advocacy opportunities? Exploring a new frontier can be pretty risky. You don't really know how it's gonna turn out. You don't actually know what's possible until you put those wheels pointed downhill towards the Colorado River. You don't know you're gonna get out of there. Next slide. In the Grand Canyon, there was definitely risk of death or bodily harm, even though I feel like we felt that this was pretty well mitigated with the team that we had. It was incredibly vulnerable, though. Headed downhill first is usually not the best strategy when you're reliant on this machine that has so many opportunities for breakdown. You kind of, go uphill first, and if stuff breaks, then at least you've got gravity on your side. I mean, there's no plan B, right? Like, we're not walking out, obviously. There's risk of embarrassment when all these eyes are on you. Both Quinn and I worked in search and rescue teams for the National Park Service with regularity. Can you imagine where our heads would be if a search and rescue was initiated for our trip? Like, you can hear these murmurs now, like, what the hell were they thinking, right? They don't belong out here. Similarly, there are risks involved in the frontier of advocacy. There is that same risk of embarrassment, honestly. You don't belong here. You're not smart enough. Where is your PhD? Why are you complaining? How dare you critique me for what I'm doing for you? There's the time, the resources involved. There's the vulnerability of hope for change, just to see that hope dashed when you come short on your goals, when that committee, it doesn't pass through that committee or it doesn't make it to the floor for a vote. But the benefits of advocacy are immense. The pride that you'll feel having rolled forward to make a change, seeing that your testimony helped a critical bill pass through a committee, seeing that what you organized led to insurance coverage for critical therapy, seeing that your input for a study design improved the effectiveness and application of the work, seeing that your input helped steer millions of dollars in the right direction. Next slide. Last point, and this is the final one, and probably the biggest one. The thing that jumped out to me the most is the importance of teamwork. This might be the most obvious one. There's no way in hell that we were getting out of there, out of that valley alone. Not happening. At Unite to Fight Paralysis, we are trying to do whatever we can to support your voice, organization, and advocacy for this community. Next slide. We've got some great changes going on. The Cure Advocacy Network, which was our former program that was committed to legislation, we're broadening that idea to be more inclusive of all of our priorities at the state level. So in about an hour or so from now, when we have these breakouts during the advocacy session, Jake and I are gonna go over Unite to Fight Paralysis priorities or opportunities for advocacy. But we're also going to invite all of you folks who are representing your organizations and advocacy groups to tell us what opportunities that you have available with your groups. Next slide. Yep, that's it. Thank you so much for your time and your attention. Appreciate you guys for all coming.
Thanks, Jason. Stand next to each other. So I just want to give a brief introduction to the next session we're going to do. Because in full disclosure, I'm a little nervous about it. If you think about just the last 2 days, we spent a lot of time talking yesterday about spinal cord stimulation, and as cool as that is, as emergent as it is, I suspect it's not good enough for restoring function and the functions that many of you in the SCI community want, or like me as a parent want for my son. There are other strategies, and that's why we then talked about combination strategies. What can we imagine combining with spinal cord stimulation? What could we imagine combining with rehab, combining with any number of strategies? But one of those is regeneration. And I'll point out, you heard a word a couple times in the presentations this morning, sprouting, that there was axonal sprouting. I've read that many, many, many times. And you heard things like BBB scores and strategies to regenerate in animal models. But there's a lingering question of like, well, why aren't they working? Why aren't they crossing over? Why aren't they good enough? So we wanted to have a conversation about that. And Sasha is going to give a presentation. And then we're going to follow that with 3 researchers in this space to have this conversation and open it up for you for comments and questions. So I just want to give you that sort of background and framework. Sasha, take it away.
Okay, but I don't have a screen. Oh, okay. Oh, there we go. Okay, hi. Sasha Rabchevsky. About 5'10" when I'm lying down. Just take a ruler. About $140. Blonde, used to be blonde, now gray. Hair. White. Wearing a khaki pants and a button-down gray shirt. Some would probably identify me as being handicapped because I am using a wheelchair. I'm a T5 paraplegic. I was injured in 1985. And I was the first paraplegic I ever met when I came out of a coma. Didn't know what that term was, didn't understand anything. My father almost killed a neurosurgeon when he used the word paraplegic. But that's the introduction. Then took a lot of decisions had to be made. Was not going to be a football player. Was not going to do the things that one would wish. And I chose the route of academia. I decided to go back to school, get good grades instead of scoring points. And it led me to doing research as a graduate student at the University of Kentucky with Paul Reier. And that's where I introduced myself into a lot, and Dr. Paul Reier obviously did it for me, a lot of new research at the time. This was in the 80s. The 85 was my injury. There was no ADA in the United States. That was in 92. So it was a very interesting time. Then I did a postdoc fellowship after I got my degree in Paris, France, very accessible place back in the 90s. And it also introduced me a lot of just how to just cope with things, because sometimes no matter how loud you scream, just isn't going to happen. And how do you deal with that? Kind of breaking barriers and boundaries. And so then through that time, I then came back to the United States and joined, as I mentioned earlier, University of Kentucky, where I've now raised up to the rank of full professor, and I run my lab and do all kinds of fun things as we all do as professors. Not just teach, by the way. You think you all have heard a lot of the things we have to do. I underwent an experimental treatment back in 2002 to put in experimental implants I've mentioned, functional electrical stimulation. Right now I have the system upstairs. I don't want to do it, but I can stand for minutes at a time, full bodies, because I have electrodes in my legs and my muscles peripherally. When you saw that standing demonstration yesterday by Tracy, that was something I did in 2002, I mentioned it, but with a peripheral nerve stimulation principle, which really wasn't hypothesis-based because we know it's gonna work unless your motor neurons are not intact. And I have a spinal cord injury with spasticity. Having said that, moving forward, here we are, 2023. There's a lot of things that we've learned and done through this time. And the thing about regeneration that we constantly hear about, that I hear about all these years, and obviously it was something that I wanted to learn myself. How do you get regeneration? Because it was such a fundamentally cool principle. In essence, that's right, you can make something regrow. Well, from the classical definitions, we can see the peripheral nervous system is able to, although not perfectly, regenerate, and the central nervous system is not. We've also been introduced to the fact that there is this, as Matthew mentioned, a collateral sprouting going on, and that's in this blue area, where an intact axon near an injury also sprouts almost compensatory-like manner. And then in a more simple schematic, you have either a process in which you have true definition of regeneration, meaning cutting the axon or regrowth of that, or you could have this, like I said, alternative sprouting principle. So go back to 1985 when I'm in the hospital. This is the first papers I read. I really wanted to go back to school and learn about this. That's why I was able to graduate just about a semester, because behind my class, instead of missing a whole year, was because I wanted to go back and learn this stuff. That's how I got to Paul Reier's lab, was because of reading things like this. But then, and this is, to me, when you go back to those schematics, really the definition for us, even still today, of using a peripheral nerve graft that has growth promoting Schwann cells in it, that you've pre-degenerated, and that you could put it into a spinal cord of an adult animal, a rat, and connect the spinal cord to the brain via regrowth, i.e. regeneration of those axons. And so this has been sort of the gold standard for our understanding of regeneration. And then Martin Schwab has used this also paradigm. But again, we have to understand that all of our models are models. When we try to model, we try to mimic the human condition. This is a partial injury paradigm, which a lot of people still use. Because if you do a complete lesion, the chances of regeneration, as we know, are almost gone. But if you do partial lesions like this, you can get some sprouting and some regeneration. It's not perfect, it's not dynamic, but it is principally regeneration. Or is it? And this is where the question is going to come today. So I want to just tout a little bit of my own work back, a decade ago over. And then I studied for a while regeneration of descending tracts, but I started getting interested in autonomic dysreflexia, which we've heard a lot of discussion about, because I suffer it every day. People who here in the room, no one understand what I mean. When you drink coffee, you got to get out of here if your blood pressure starts rising. Not because, anyway, I think we all understand the conditions. However, this is just a demonstration of what we had done experimentally, which was to follow up on work by Dr. Krashikov and Larry Schramm, who did some amazing work at Johns Hopkins, electrophysiologically correlating sympathetically correlated interneurons in the spinal cord with sympathetic activity, meaning that there was a relay from colorectal distension or peeing and things of this nature that was triggering the system. And nobody had really known that the sympathetic circuitry was involved. So we did some tracing. Dr. Shel Pighow, my former postdoc, did some tracing. And these are not terribly complicated slides, but what you want to see here is in this enlargement, is that on the animals that got transected, which is non-transected normal animals, you can see some tracing, but look at the amount of tracing that happens after an injury when you're injecting at the lower part, not the upper part. In other words, these are sprouting, regenerating, if you want to call them, things that are going ascending ways. And I had never seen this. I was actually quite surprised. I didn't believe, actually. It made them redo it. But we actually, this was the first demonstration histologically that we could correlate that. So again, we believe though, and this is upside down for a reason, okay, that it's more like this. It's a compensatory sprouting. We don't necessarily believe it's regeneration because we cut the axons all the way high, and those are all coming from lower areas. So we think it's sort of like we want information and it's trying to get up there to seek it, okay? And so serendipitously, I don't have the slides here, but there's a lot of CFOS activation. The cells are alive. When you stimulate them, there's a lot of stuff going on in the cell, electrically, if you will, that is not normally happening in a normal cord because there's descending control. Normally it's quiet. Well, it's been unleashed. And so this was, again, this to me was fascinating. Now the field understands or starting to follow this principle. But let's go back to the term regeneration. So I just did this a couple, nights ago, and looking at just the term, and you don't have to go, I'm not going to go through each one of these, but the fact is it's a quagmire. When you go down all of these, so what exactly the species, the product, what you're looking at, and then parenthetically, when you see these quotes here, this is because these are quotes that basically you hear, and you're like, wait a minute, adult nerve cells, and isn't it a nerve in the spot? Isn't it out of the spinal cord? Isn't it in the periphery? And so there's a lot of kind of like lingo that is not, in my estimation, informative, because I've seen regeneration, as many of we have here, for decades in titles, but yet we don't really have it. Well, we have indices of it, but we don't have now what is the definition of regeneration. That's sort of what we're going to be discussing today amongst panelists that have expertise in these fields. So I want to just stop for a second and remind people that this guy up here on the screen really pissed off a lot of people. I don't know if you all remember this. This is 2020. And in essence, he was chastised by quite a few people, and I lauded him, okay? And why? It's because, again, if you look at what Vance Lemon had surmised, was that Barry was saying that scientists are working on the wrong problems, there's a lack of sense of urgency, but everybody remembers, he says, we're dying here, right? Come on, he wasn't telling you, the investigators, that you are killing us. That's not the intent. It's just to raise awareness that as you all are going through your 15, 20 years grant cycles, we are dying. Are we seeing these repeated patterns all the time? We see the same things going on in press and in hype. And so with regeneration, I'm quite tired of seeing regeneration and reading the papers and telling my students it's not really regeneration. In essence, how do you do that? If we as the stewards of this field are supposed to be leading people, why are we going into this area where, like, well, in that lab they kind of call it that, in that lab they call it that, and the journals accept it. So the other thing is the journals need to step up. I think we've been talking about this. Now, before I introduce the panelists, excuse me, this is the quote. You don't have to read the whole thing. And actually, we will just be discussing this more later. But again, we need to figure out how to really disrupt things, is what Vance Lemon surmised. We need a different way. We need to be doing things much differently, because seeing as you guys are complaining about it and we're doing everything we can, you all come up with the idea, which was really, to me, a very, if I could feel them a kicking somewhere, that's what it felt like. Okay, now, how can we fix science came up in a discussion where we had the International Spinal Society for Neural Regeneration just last April. And there was a lot of discussion about it, a lot of opinions, mostly obviously scientists, because this was not really a venue for people with this lived experience. But the funding, replication, translation, and mechanism, I think Matthew kind of liked to use that term, fetish for mechanism. And I understand what that means from my wheelchair standpoint. The fetish of mechanism, you're like, I don't care how it works, just give me some. Right? But that's naive to think that we can just use you all as guinea pigs and that we can just say, sure, try this, without really understanding the risks that are involved in delivering these things to you. And that's why everybody here is, including myself, a little bit cautious with what you say, because you don't want to make aliens of people that you hope are going to help you along the way. So with that, I also want, this is what I was going to suggest, is that this just came out in Nature commentary. I think that publishers that are allowing publications to use these, as I'm going to call it, hype, why is it that they're allowing that? Well, I think we all know. Is that correct? Is that really what we should be doing? Because we as scientists are looking at them saying, please take our papers. But really, is that how, I mean, you know, I didn't get accepted for 4 rounds. Well, did you do anything better? Yes, we addressed things. But again, there's a quandary of why is it that there's a hierarchy? So I would like, oops, sorry. So I don't believe, and you can read up here, that it's UTFP's mission to come up and solve people's problems. We are already out there trying to explain what the problems are. And when we get these things like, well, fine, you figure it out, it really is a slap in the face sometimes, to be honest with you. But that's okay. I kind of endear that because it's always been, at least in my circumstances, the way, because it just doesn't have people we use the word ableism, and I've discussed with many people with that. I'm not going to sit here and say what ableism is and what it isn't, but deep down inside, I think we all know with the C-, D grades, we're not passing the tests. Okay. And that's everybody, introspectively, myself as well. And I think that once we all can draw that guard down, if we can possibly do it, I think there's going to be a lot more dialect and more sharing. We've already discussed data sharing and information, but more willingly sharing because, oh, I met the people at this meeting and they were at this other meeting, because since I've joined Unite to Fight Paralysis in 2017, my horizons have extremely broadened because it's brought me to a different audience and it's brought different people to hear my and me their experiences. Clinicians and scientists as well, by the way. Okay. For a long time, I was the only one in attendance at meetings. I'm not joking. Okay. We're talking 15, 20 years. And it would typically be me and on committees because why? We needed a disabled person on the committee. I'm not saying that's bad, and I did it, but you know, really, that's how fast. Now I'm looking at the room. I never would have expected this even 10 years ago to see this many people who live experience around. Okay, and it's wonderful. And Jason and I are good friends now because I started off as a technician before I started graduate school when I was paraplegic. So I mean, it's a nice kind of a tour de force. So this is the questions. If you can read it here, let's not take any more time because I want to introduce Parr, Murray Blackmore, and Ricardo Battaglino to come up stage. And we're going to basically, if you look at here, read up here, we're going to have three principal questions we'll be addressing. We will then stop, and once they answer these questions or address them, we'll open up the floor, and then we'll go to the next question. So please, Anne, and Ricardo, and Murray. Could you keep the screen on for the next slide, please? Thanks. I guess, can you all just introduce yourselves briefly?
Yeah, I'm Anne Parr. I'm a neurosurgeon at the University of Minnesota, and I also run a spinal cord injury lab. We do some studies on epidural stimulation as well as stem cell transplantation, and we focus on regionally specific spinal cord neural stem cells that are derived from iPS cells or induced pluripotent stem cells.
My name is Ricardo Battaglino from the University of Minnesota Department of PMR. And I do better like that. Yeah. I'm a preclinical and also a clinical researcher. I've been working for many years with rodent models and also in human studies, spinal cord injury.
Yeah, hi, I'm Murray Blackmore. I describe myself as a pretty basic researcher, rodent models, and striving for regeneration using different approaches.
Well, thank you for introductions and for being willing to come out and do this. I know this is kind of like a loaded gun questions, you know, and I think there's a lot of people, Matthew was asking, like, what are you going to do? What are you going to ask them? Ann and I discussed it, all Murray and I have. This is very open. I want to be very, very casual about this because I think that when we all go and talk afterwards, when we're drinking, I think the discussions will go on. And there's another session after this, actually, in the afternoon. So in the words, you've heard what I've discussed. Each one of you, I'd just like you to make some general comments about regeneration. And you've worked on regeneration in preclinical models. You transplant in human beings. Could you just give us, and then Ricardo, and then Murray, please.
Sure. Yes. Thank you very much. Am I talking loud enough? Everyone can hear me. Thank you. Yeah. Thanks so much for inviting me here today. I'm not sure why, Matthew, you were nervous about this session. I think this will be fine. And I mean, I'm always willing to talk and be honest about, my opinions about this. I think when we talk about regeneration and the focus on, what is regeneration and the definition of that, to me, I'm sort of like a very black and white person in some ways when I explain things, because I really like people that understand very simplistically, and maybe I'm just simplistic. But the first thing I do when I'm explaining about, for example, mechanisms of cell transplantation, is that I say the most important thing I think is to understand the difference between neuroprotection and not neuroprotection. And that is actually one of my main focuses, because I think that for the most part, and this is also debatable, but neuroprotection strategies are really probably only going to work in acute or subacute spinal cord injury and not chronic spinal cord injury. And so, I've also heard the argument, people that want to advocate for neuroprotective strategies, I think they're great. But they say, oh, if we can just get a good neuroprotective strategy, then we'll just cure spinal cord injury and nobody will have spinal cord injury to begin with. And we don't need to focus on chronic injuries. But I think that that's really a fallacy, because first of all, not everyone is going to get the treatment up front for a number of reasons. And then what about all the people that are out there now with chronic spinal cord injuries? And so I think that's really important. And, how many times have I read a paper or something that says, okay, the mechanism is this and regeneration and axonal regeneration and protection and this and this. And I just think that to not focus on one thing or the other is really missing the point, because I think one of the main things is that you have to know whether this is likely to work for chronic spinal cord injury or not. And like I say, neuroprotective strategies are great, and I think it's great to pursue that, but I think you should know that up front. And so if you're going for a chronic injury, I think you have to go for a regenerative strategy. And that's how I see things, but how you guys see things.
Okay, regeneration. It's a fine term. It's actually a really simple term. The problem with it is how little information it carries. So axon regeneration, what it means is that it's growth from an axon that got disrupted, that got cut, right? As opposed to growth coming from an axon that didn't get cut and it's just kind of sprouting. So when you hear the word regeneration, you only know one thing. You know that a cut axon is growing. Now, the problems come in because you need to build information around that term to make sense of it. And some of it's scientific, but some of it's just common sense. And I'm going to just lay it out. The first thing when you hear, okay, so regeneration happened. Great. How far did it go? That's the first question. And you can do that in distance, but that's a little hard to make sense of. So I like to think in terms of segments, right? Like the spinal cord is segmentally organized. So is that growth stuck in the segment of the injury, like within segment regeneration? Did it make it one segment down? Did it make it many segments down? So I'd like to sort of classify it within single or multi-segment regeneration, right? That's one thing that you would like to know. The other is, it's easily missed. It's what % of the axons are regenerating, right? So you applied a treatment, did 2% grow? Was it 3%? Was it 50%? What was it, right? So those are the big two in my mind. And then there's these kind of like science-y side points, like where on the cut axon did the growth come from? Is it from the end or from the edge? Is it kind of arborizing and making connections, or is it growing a long distance? And then there's the which axons is it, right? There's many different types of axons in the spinal cord. They're going to carry different functions. So you kind of need to know which population you're talking about. So here, let me make this a little more concrete for you, though. You'll read two papers. Treatment X caused regeneration. Treatment Y caused regeneration. And they're both going to say robust regeneration, because that's what we do. It's a meaningless term, right? It just means good at this point. We had good regeneration. We had robust regeneration. But if you would say treatment X produced within segment regeneration from 3% of axons in a highly arborized connecting phenotype, okay. Treatment Y produced multi-segment regeneration from 20% of axons with a linear extending phenotype. Okay, now great. Now you have the information you need to start to make some judgments about which is important and which isn't. And so just to summarize, my position is that the problem isn't the term regeneration. It's a really clear, I think, well-defined, accepted term. It's the failure to build around it the clear information that we need to distinguish and evaluate the different types of regeneration. And I think there's systemic reasons for that failure. I'm not sure I'll leave it there. I think there's reasons why that's the case, but I've said my piece.
I think I have a question for you. So when we talk about regeneration versus sprouting, the the basic cell biology behind those two processes are fundamentally different, or sprouting is just like
Compensatory.
like a smaller scale regeneration process.
This is what Sasha's talking about, the way that the terminology can make it so hard to even communicate. So here's the problem with that question. He's asking, well, is there a difference in the mechanisms by which sprouting happens or regeneration happens? But first you have to say, well, what do you mean by sprouting? And the trouble is there's been sort of two definitions of that term. Because scientists have been wrestling with this for a long time. So a paper was published that said, okay, regeneration means the growth from an injured axon. That's simple enough. Sprouting means growth from an uninjured axon. Okay, so that's one definition. But then sprouting can also just be a description of the manner in which an axon is growing. Like, is it growing in a straight line, or is it kind of sprouting all over the place? And that's how another set of scientists use the term sprouting. So in order to answer the question, how are you using the word sprouting? Do you mean specifically from an uninjured, or do you just mean the manner in which the axon is growing?
No, I was thinking about growth from an axon that is uninjured and growth from an axon that has been injured. If those two phenomena were fundamentally different. I
understand the question. Well, it's hard to say. They're probably, well, okay. I think they're different, but it's not the important difference. The important difference, and this is where, I don't know how like sciencey and inside baseball we want to get, but I think this has created huge confusion in the field, so I'm going to say it. Whether they're injured or not, axons have the option to grow in two very different modes. One is a short distance arborized mode that's designed to make connections. The other is this long distance straight mode that's what you need to cover multi-segment. What the field has done is to take pictures like within a spinal cord segment of this really arborized mode and said regeneration has happened, we've succeeded, that's regeneration. What the field hasn't done is really convincingly made that long distance mode happen. And so that's where I divide things. There's an arborizing mode and a long distance mode. And as far as I can tell, our therapies have done a really good job of making more of that short distance arborizing. They have not succeeded in that long distance mode. And so, yes, I think it's two different mechanisms. And yes, I think one of them is still a frontier and a basic stumbling block.
Okay. Not here. Hello. Okay. Sorry. I want to turn it around because you had used it. Oh, there it is. You had used the word regenerative medicine or something to that effect. That's what I sometimes, when I looked up and I had the term up there, when you think of regenerative medicine in the literature, it's stem cells. It's not necessarily regeneration. It's regenerative cells that then can extend axons. So that's what I wanted to ask. When you have an axon, a cell that's in there and extends an axon, do you consider that when you trace that, is that regeneration or is that just growth? Because it's a new cell that is putting a process, so it's not regenerating per se, it's generating. No, again, I'm just trying to understand it because it's the first thing when you do a Google search, it says stem cells, but that's the first thing though.
Yeah, that's an interesting question. I think that it's a great panel because we all have very different perspectives, and I don't disagree with anything that you're saying, but I just have a different overall, the way that I approach things. And I think it's the clinician in me and this top-down approach. So again, I hate to come back to really patient-specific therapies, but to me, I ask less the question, is this sprouting or is this considered regeneration? Because to me, it's a lot of semantics. I more ask the question, okay, you have a patient in front of you, what's going to be the most helpful and what isn't, right? And so with a cervical patient, we know that if you can get some sprouting with a cervical patient, you can get, as you said, if we know we can get 1 level, 2 levels, 3 levels, that's meaningful, right? In a thoracic patient, it's not meaningful. All you're going to get is a couple more levels of sensation. And so that's kind of I think my response to that. Like I say, I don't disagree with anything you're saying. I kind of look at it from a very different perspective. I asked the first question, like, what is our goal here? What is the mechanism that is most likely to help this patient? And then kind of focus from there. As to your question about regeneration, I guess I use the term regenerative medicine very loosely too. I use it to mean pretty much anything that's not neuroprotective. I really do. And even like epidural stimulation, which we do some of that. I consider, so one of the things that we've discovered, and Dr. Darrow and Dr. Samadani have worked with me on this as well, and they've been key in those trials. They've shown that after turning off the stimulator, there are patients that still have maintained improvements, functional improvements. So we ask, what is that? And I'm like, well, there has to be some neuroplasticity involved there. And I'm like, so we've done some epidural stimulation in rats, and we've discovered a few different things. One is that activated nerves cause remyelination or cause increased myelin. I shouldn't say remyelination, but cause increased myelin in the area. And so we know that's one mechanism. Is that regeneration of potential oligodendrocytes, which is a whole different thing? Maybe. So I would loosely use the term regenerative there. Is it from sprouting? Because we've shown some increase in synaptophysin in the area. So there's probably some synaptic plasticity. And we've shown increased serotonin, which is also known to be highly involved in spinal cord function. And so those are all potential different mechanisms. I would say increased serotonin may not be regenerative, but we don't know where that serotonin is coming from. Is it possible that the synaptophysin is increased synapses being formed from local sprouting in the area? We don't know. Could be regeneration, and also increased oligodendrocytes causing increased myelination. Do we know if that's regeneration? I use the term loosely. I recognize that other people don't. I certainly would not say that all regenerative medicine means stem cells. I would not even.
No, I did that.
Yeah,
for both
yeah, no,
because you're
absolutely,
stem cell
absolutely.
biologist, yeah.
Yeah, and so that's kind of my take on it, but I think that sometimes we get down into the weeds a little bit too much, maybe, arguing the semantics of things. And I would, like I say, I'd rather take sort of more top-down approach to what do patients need, which mechanism would match the best with this patient's injury, and how can we achieve that?
Right. So having said that, and we go on to some other questions, is that we have this new advent of stimulation, which is just blossoming and blooming. And for a long time, as I understand, you were doing the regenerative approaches, and now you're doing stimulation. Do you see where, like, obviously stimulation is not going to do everything, right? I mean, we're seeing that. Do you have any particular models that you would show, and I'm asking all of the panelists in a way, that would use, hey, we know this regeneration model works in an animal. Let's see if giving it stimulation increases that. And if you'd like to kind of talk about that, possibly. I don't know if that's too much into your CDs, but is that possible to think that way? Anyway, yeah, Murray?
Okay, trying to organize my thoughts. Okay, first of all, it's not semantics. It's not small scientific thinking. I'm coming from a very top-down perspective, which is what do we need to do that we don't know how to do right now to cure this injury, to restore near normal function to most people with SCI. That's my North Star. And in my humble opinion, you cannot sprout your way to that or anything very close to that, right? So it matters how the axons are growing, because you can't sprout your way more than you can't. What you were saying, you just casually threw in like, oh, a cervical individual, you can sort of sprout your way down a few segments. No, sprouting covers 1 or 2 millimeters, maybe 3 if you're pushing it. And that's not going to get you multi-segment. And so I'll leave it there, but I just want to make it crystal clear that when I say there's this major problem that the field hasn't solved yet, which is how to get axons to grow multiple segments and make connections, it's still there. And if you hear frustration in my voice, it's because by conflating these terms, by using regeneration to kind of mean everything, this idea comes, which was baked into that question. Well, you know, things are working. We've got regeneration in animals. Why isn't it translating? What I'm telling you is we don't. When you look quantitatively, really carefully at the animal data, we haven't done in animals what we would need to do in order to reasonably expect a lot of function in humans. And that's a strong statement for my field, but that's actually what I believe. So I don't think it's a model question. I think it's like, what are you measuring in the model, and how clear-minded are you about if the axons have really gone far enough to make the difference that you would hope they would? Thank you, Mark. Ricardo? Or Ann?
I was just gonna say, I don't think we're saying different things. We're just saying them using different language, because I don't disagree with anything that you're saying. I guess my point about the cervical injury wasn't about sprouting per se. Maybe I casually threw that word in. What I just meant is that, like, might be successful in a cervical injury is going to be different, or some of the things might be the same, but my point was just that patient-specific treatments need to be found, because what is going to work or give somebody benefits is not going to work for everyone. For example, epidural stimulation, autonomic dysfunction. Well, a lot of people with high cervical injuries, high thoracic injuries have autonomic dysfunction. People with low thoracic injuries tend to have less autonomic dysreflexia. So if you put in epidural stimulation, looking for specifically blood pressure control, might be different for different patients. That's all my point was.
Yeah. Oh. Ricardo,
No, what I'm thinking is that, just echoing on what Dr. Murray here was saying, I think it would be important for the field, at least in preclinical studies, to basically increase the magnification on this phenomenon of regeneration, and basically do what we do in any other field, just put numbers to it, quantify. So Murray was just saying precisely, well, there are different things that we can quantify. For instance, the extent or the length of the growth of that axon, and how many of those axons are doing that. I mean, that's exactly what we do in any other situation. We need, and I believe that if we were to do that and then reanalyze a lot of this data, we will find that some of the things that apparently had similar effects just by measuring, let's say, the our BMS scores and measures of locomotor recovery might end up being quite different. So I think that will, I'm thinking about now how in many other areas of research, researchers are trying to agree with each other on, what is a biomarker? What is the level of certain biomarker that will, be prognostic or diagnostic of some condition? And this is the same. I think that Mary is trying to shed light on why don't we all agree on what we're going to call what, and then the literature will start making some more sense, probably. I mean, and I haven't heard something like that before. I think it's very interesting that it's basically like increasing the resolution on this phenomenon of regeneration and try to look deeper inside and see, well, what are we actually achieving at the cellular level, at the histological level? And I understand that may not be necessarily what the way we think about this in human studies, but I think it will be extremely important, because otherwise this axonal regeneration becomes like a black box. Something that we don't know really what's going on in there, and just we put in something on this side and we see an effect on this other side. But if we are going to understand how this is working with the hopes of maybe helping it or, stimulating it or inhibiting it, we will have to understand more clearly how this is working. And,
sure. And that's what this panel's about. So
just demonstrating just the complexity of what's going on. And now, Matthew, I think you have.
Yeah, I just wanted to make a quick comment that, my nervousness, Ann, about this is not so much that anything crazy was going to be said, but without sounding too relativistic, I think there's a part of this is that we weren't trying to answer a question like with a truth or a dogma. It's really, and I think in some ways, I heard it resound very loudly, the breadth, both the breadth and the tension between Ann, the way you answered the question, and Murray, the way you answered the question. There is and how we in the community rest in between that, right? You use Barry's, we're all dying here. Like the next time we have a decade of disruption, I might be dead when we come back, right? So there's a pragmatism here to like, well, what's gonna matter? And it was a question I was gonna throw at you, Murray, around like, well, what is regenerating? What kind of cells and what meaning have they? What function have they? That's the other part of the question, Ann, that I think you brought in. It's like, well, okay, we did this regeneration, we had growth, but so what? What did it do? What will it do? Give me sensation back, give me mobility back, give me more pain? Any of those sorts of things. And so with that, I just wanted to we have a question from the live stream from Paulo. Do you think that in the case of chronic SCI, to turn on the regeneration program of injured neurons will be enough to get functional recovery? Or first, will it be necessary to make the injury site regeneration friendly and provide proper axonal guidance signals toward proper targets, meaning long distance regeneration? And then let's open it up for questions.
I don't know who wants to take that first.
It was addressed to Murray.
Oh, okay. Good. Yeah, to cure spinal cord injury, you're going to have to do a few things, right? You're going to have to put some kind of a bridge in the big cavity in the middle of the injury, because you can't grow axons over nothing. So there's going to be some bridging component. Baked into that, there might be some, you might have to address the barriers at either side of the bridge. Right? You're going to need to stimulate the neurons to initiate long distance axon growth, and that's what I work on. And then there's going to be this whole process of kind of pruning and refining those connections, and that's where epidural stim and PT is going to come in, right? So you've got these three major pieces, and they all have to work together. So no, long distance regeneration by itself, that's not going to completely functionally cure spinal cord injury. Neither will bridging, neither will the sort of epidural stim PT side of things. But they have to work together in a coordinated way for sure.
Something that I didn't mention during my presentation was that when I saw this regenerative sprouting or sprouting of the intact axons going up, was that there's a lot of things that it's connecting to that it shouldn't be, right? When it's going up, it's connecting to a lot of connections, but they're not supposed to be making those connections. As a matter of fact, that's the basis of the development of autonomic dysreflexia. We don't have time to go into the mechanisms, but if you thwart that, at least according to recent work in animal models, you can prevent some of these conditions of secondary complications of autonomic dysreflexia. So having said that, then I think, Murray, I want to make sure that people understand something. This man is dedicated to this field, and he takes a lot of brunt because he's not willing to hold back the tough questions. And he himself is his worst critic. And I don't want to give him applaud right now, but buy him a drink later on, okay? And I mean this, Murray. Murray has his own vested interest in why he's into this field, okay? He has people with spinal cord injury in his lab. So this is not somebody who doesn't thwart the questions, okay? But he doesn't have the answers. And as you saw yourself, he has one because his goal is to understand the mechanistic way and is to make people better. That's a big difference, right? That's a very large difference in approaches. It doesn't mean they're not talking the same language. It means that we all are learning each other's disciplines. It doesn't mean that we're correcting, but at least appreciating the whole spectrum of these different silos that we keep hearing about, how we can listen to each other and learn. So I don't know how, but do we have any more questions?
Yeah, we have some questions. Isabel.
Isabel Scaresbrook from the Mayo Clinic. Thank you for this really important conversation. And it's great to see a lot of different ideas all circling around the idea of regeneration. First, I just wanted to make a quick comment on regenerative medicine. I think the definition could be all over the place, but generally speaking, I think it's accepted that regenerative medicine is when you're trying to restore form and function. And whatever it takes, and you restore form and function, we can call that regenerative medicine. Whether it's the spinal cord, the liver, the heart, we want to regenerate and restore function. So the other question I had is when you think about a spinal cord injury, particularly a chronic spinal cord injury, it's still a very active environment. And so I'm wondering, maybe Anne or others can address it, can you really separate neuroprotection from regeneration? Because if we are able to regenerate axons, do we still need some neuroprotection to protect them from the ongoing onslaught of what might be going on in individual spinal cords? Thank you.
Yes, thank you for the question. I was kind of avoiding that a little bit because I like to kind of, just for the sake of simplicity, put things in different boxes. But there is some evidence that perhaps there is a long-standing immune response against the spinal cord after injury that may, if you can find something, and I don't really think of that as regeneration necessarily. It's more like prevention of deterioration. And so I didn't really get into that for like I say, for the sake of complexity, but as a clinician, I do actually see patients who are long-standing spinal cord injury patients in my clinic who are deteriorating. And we don't really know why. Usually they get sent to me because they get a diagnosis of syringomyelia and there's no evidence. It's usually just myelomalacia. It's chronic, long-standing, but I don't know whether it's because of long-standing inflammation, which is one theory, or whether it's because, unfortunately, sorry, it's probably long-winded, but unfortunately all of our brains, we know, right, start deteriorating when we're like 30, but we don't notice it until we're much, much older. And the reason that's true, and the reason that we don't notice it is because we have redundant systems, and your redundant system, so you have a lot of redundancy in your brain that can manifest until you're much, much older. And then you start noticing the dementia kicking in, unfortunately. I think that the spinal cord, I've always argued the spinal cord is another lobe of the brain. I think it is. And I think that the same mechanisms that apply to the brain often apply to the spinal cord. And it's often people that are a bit young for dementia, but kind of around 50, 60 after a spinal cord injury that do sometimes come to my clinic and manifest with unknown deterioration. And again, this may be something, so it's certainly a worthwhile area of investigation, whether it's chronic inflammation or whether it's just, unfortunately, that part of the natural aging process. This is something I've always wondered, but I haven't researched myself.
Yes, question?
Sam?
Is this working?
Yep.
My name is Samantha Troyer, and in my investigation to get an epidural stimulator, which I had implanted in February, they talked about how the epidural stimulator can change the structure of the spinal cord. And is that correlated to like the regenerative axonal and sprouting that they're referring to? Or is there like a different process going on from the epidural stimulator changing the structure of the spinal cord?
Okay, epidural stimulation can affect like short distance growth of axons. The substrate for that, the axons that we're talking about though, would be the axons either that I apologize. I'll cut to the chase. It's not regeneration because we're not probably talking about the growth of the injured axons. It's more sort of new growth and modeling in the spared circuitry and the circuitry that's down in your lower spinal cord. So yes, new growth, but by the strict definition I've laid out, probably not regenerative growth. But yeah, that's one man's opinion.
Yeah, no, by your definition of regeneration, then I would agree with that. But I kind of laid out some of the possible mechanisms that we've identified in rats, and we are back translating this to a pig model as well, so that we can look a little bit more in depth about actually why that happens, because we don't really know.
So what Dr. Parr was describing was that some of the effects, at least as I interpreted, of the not necessarily making regeneration of axons, but the circuitry exists, is making them myelinated so they are more efficient, or if they're dormant, they wake up because they're newly myelinated and they can conduct. Is that correct? Am I speaking out of, okay. But just
Well,
for those who didn't understand that, yeah,
I
sorry.
was just going to add that, I mean, since Ann brought that up, some of you know, maybe a lot of you don't, that we cooked up an idea in conversation with some of our partners, Ann being one of them, and you know about the nerve gen peptide. And again, thinking about combination, and in a sense, really asking this question that Paolo asked, like, do you need a more permissive environment to amp up what spinal stimulation is kind of maybe hypothesized to do, right? To take these spared signals and amp them up, or sort of utilize them for more function. So we cooked up this idea and approached Jerry Silver and NerveGen and Candace Floyd and Ann Parr and David Darrow and said, hey, could we combine the peptide that's being studied in humans with an epidural stimulator that's being studied in humans in a chronic pig model, a large animal that Igor was talking about earlier, better than a rat or mouse, at least anatomically. Can we put them together and see what happens? We had trouble funding that in a conventional way. So 6 spinal cord injury foundations, some of whom are in the room today, said, well, we'll do it because we think this is valuable to answer that question. And it gets back to what Paolo said about, I mean, again, for us, not scientific, it's more like common sense. Like, can you make a more permissive environment? At least, again, hypothesizing that the peptide will do that. And then you amp it up with this neuroplastic inducing spinal stimulation, can see what we can do. And we'll find out. That study has started. It's in its early stages, and it's going to take a little while, but we'll be reporting on it. Jason, question. And then we'll go to Marco, and then we'll come over here to Susan.
Jason with U2FP. Hey, I'm hoping that you guys can help clarify a question I have about the pathophysiology of the cord after the trauma of spinal cord injury. So the axons distal to the lesion, I've heard different things about what's going on there. I've read that within 3 days, there's quite a bit of degeneration occurring in those axons downstream, or atrophy. But it also seems kind of confusing because the work that's being done in neurostim and neuromodulation seems to show that there's some activation of signal getting to peripheral nerves to activate motor function to get legs moving. So what's going on? What's going on with these distal axons? Are they recoverable? Can we connect to them again? Also, like, how are we gonna, if we do get through the lesion, somehow, some scaffold or however we're gonna do it, how are we gonna make those connections? Thanks.
Can I start? I'll start, then you all can. As I understand it, you talk about the distal axons below the injury site. We can't rescue those. They're gonna die back. So that's pretty much this. I don't know how much more you want to get into that, but we cannot rescue those that have been damaged and are retracting. It's called Wallerian degeneration. It comes up to the injury site, and then after that, there could even be further degeneration. And we're hoping that that connection above can be regenerative, that we could take that degenerating and rescue it and sort of go back. But that's the challenge. Below the injury, I'd like some comments. I don't believe you can attach that.
So just clarify really quickly, Sasha. So then the signal that's somehow getting through with like neuromodulation to activate these motor neurons, are those peripheral nerves that are leaving, or root nerves that are leaving, that are being activated through that?
No, I think this goes back to answer the spinal cord, as I demonstrated, when you cut it, it's still active, it's alive, it's got input, it's got output, and you can stimulate that without having any cortical control. You can do that by demonstration of FES, or even like what was demonstrated on the stages here yesterday. So I think it's just you have every segment has input. Here, input. It doesn't mean that you can feel that after you have an injury like myself, below that, but it's still activating, which is why you have reflexes unless you have motor neuron, a lower motor neuron injury or disease. So I'm trying to not dismiss your question. I'm just trying to figure out what your misunderstanding is. But say that, Naomi, please. Could somebody, yeah. Well, therefore, this is what the audience is about, Naomi. Fine, here's the expert.
So everything Sasha said was exactly right. You have to think of one axon. If you cut it, that part that's away on the other side dies back. It's not connected to the cell body. It's like if you have an amputation, your hand won't survive. But it's as if you had 1,000 arms. And so there may be a few of those axons that survived and went past. And those are the ones that may be changing in exactly the question you're asking. But it's very different than the rest of the discussion there. And then what Sasha's saying is there's a whole bunch of other cells down below that waiting for input. And he's right about that too, but you're just missing that one connection that there might be some of those nerve cells that made it past the injury and are not degenerating.
Marco.
But Marco Battista, Christopher and Dana Reed Foundation. When I was in industry, we would replicate everything that we did. So every new program, we would have replication studies, and it was shocking to me how difficult it was to replicate a lot of work. Curious on your thoughts on the state of the field for preclinical regeneration with regards to replication, and what if it's not doing well, what are the possible solutions around getting better independent replication?
I'll let you guys take this.
Well, it may go back to a certain extent what Mary was talking about before. I mean, before I was working with spinal cord injury, I was a bone researcher actually for many years. And just like in many other fields, we had a number of standard measures that we had all agreed on. And so it was easier to compare literature in many cases because we were comparing numbers that were obtained in a similar fashion, and then you could take two studies like you're saying. But that also happens in spinal cord injury study when we talk about locomotor recovery. There's a number of measures like the BMS score and things like that. So that allows us to compare, but that doesn't seem to be the case when it comes to precisely I wouldn't say regeneration, the regenerative ability or capacity of either interventions or essentially, drugs, proteins, stem cells. So we, again, the system seems to be treated a little bit like a black box. We introduce an intervention on one side. We inject the protein cells, but then what we measure, the output is something that is very far removed, and we are not quite sure what's going on in between. So until we agree on a number of parameters and numbers that we can literally measure and compare to one another, then we won't be able to compare literally, and that may be why it's so difficult to reproduce some of these studies. Somebody had asked before, I forget, with regards to dieback of axons at the injury site. There are some reports in rodent studies that look at precisely proteins that may prevent that. That can be measured too. So I'm not 100% sure that all those axons that have been injured cannot be recovered after the acute phase. But it may be, again, a question of numbers. How many of those axons do you need to not die back and then cross the injury site? And this is, again, going back to Mary was saying, we need to be able to put numbers to this type of outcome. Otherwise, we just relying on, or we mostly relying on functional measures that may I mean, we may get to functional recovery or to partial functional recovery, especially in rodents, by many ways. And it may be a combination of sprouting and regeneration. But generally speaking, like you said, I mean, it's been hard to achieve in rodent models, long distance regeneration. Yeah.
Yeah, real quick on the replication side. I mean, there's a history here. Many of you know it, but for those of you that don't, the NIH funded replication studies. The whole idea was to take the most promising SCI findings, preclinical, and then just have another lab repeat them. I think Naomi would remember this score better than me, but by my memory, I think it was 2 of 11-ish, something like that. What's that?
1.5.
1.5 of more than 10. So there's a major replication problem in the field. I don't think it's been resolved. We haven't, as far as I know, there's been no major effort since that time, which might have wrapped up 8-ish years ago. So let's just acknowledge it. I think that's still a lingering problem in the field.
And I think it's frustrating from my standpoint personally to see all these years go by and we're getting guided by funding agencies of what we're supposed to do. And yet there's no mechanism of accountability. What I mean by that is that, okay, yeah, you got this paper, now you're gonna get another grant, but there's no accountability for what was stated many times. And it gets buried away, you never hear about it. Well, what happened? Oh yeah, we didn't have the funding, or it didn't work, or it wasn't powerful enough, but yet the field is hearing that, right? That's, to me, again, I'm not gonna be a protagonist here of beating down the publishers, but there is a certain frustration on many people's part in this room of how certain things get into journals that are very high impact, that when we do them in Journal Club, we tear them apart. I mean, tear them apart with graduate students. That's not fun, because you're sitting there and the students start beginning to ask, is anything without flaw? I mean, we have a specific seminar series to say, you find Waldo, right? Because there are critical flaws to things that are published, and maybe there's a retraction, maybe there's a statement, but it's out there. You've unleashed it, and there's all kinds of videos and stuff to go with it, and the universities in particular are sitting there publishing, this is what we discovered. Then when you go talk with the people, they can't answer. They don't have a response, or the study never panned out. Well, yeah, but you got a lot of money for all that stuff. What are you gonna do with it? What did you do with it? And that's something that Matthew was involved with, the UTFP down the road, and that's ongoing, is we're gonna really bring up the numbers of these repetition studies and all these. How much money have we invested in this versus what we've gotten out of it? And I think we all know what the grade we've got for that. It is not controversial to say this, folks. It is reality. And I'm tired of people being so sensitive of, well, that's not my boat. Well, it is your boat. If you're professing this and you're using spinal cord injury condition as a rare condition that is orphaned, use it with a little bit more integrity instead of just using it for hype. And ask yourselves in your own lab. I don't know how many people do. My lab, if you don't show a functional connection, it ain't regeneration. It's sprouting. And that's just me, because I'm a hard ass and I don't publish a lot because I can't get the regeneration that everybody else is saying. Yeah. Okay. I do the same thing. I'm off my target. So that's sorry.
No, that's okay. I just wanted to add one little piece because we talked about it and we're not going to get to it because we only have a couple minutes. We're not going to get to it in this conversation, but just to put this nugget in your head, one of the things we talked about and we wanted to talk about are, and you've heard, if you've heard us on the podcast, you've heard, I think I've even said it a couple of times throughout the last 2 days, some of these are about system incentives. So there's a reason why people write their research in a certain way to hype it, to get to a high impact journal, because there's money involved and there's status involved and there's reputation involved and there's influence involved. And it's really important to point out that there's no villain here.
Right.
We're all inhabiting a system and a structure that incentivizes you to certain things. And, you know, Sasha put that picture up of Barry. I was at that meeting and I said something similar to this. There's a lot of people in the room that did not like what I said because they misunderstood that I was villainizing people. But I wasn't. It was like we're all living in a certain house, and the house has certain paths that are well worn, and it's easy to go down them. And sometimes going off that path to try to change that system is really difficult, right? Because everybody is used to walking this path, not to go too generalistic or, you know, make it too generalized. But that's a really important feature of this that I think is important, coming all the way back to the semantics, how we understand it, what our roles are, and what our targets are, because they are a part of that incentive structure. And it's also why it's so important for us to be together and be in discourse together and be willing to listen to things we may not want to hear, including us in the community, right? So I just want to point that out. And then one, maybe we should we be done? My people? Sorry for those of you who had questions, because we're already 10 minutes over. Thank you, all of you, each of you.
Thank you
Please.
all. Thank you very much.
And one thing I didn't quite finish my thought earlier is that this was not intended to answer the question.
What?
This was intended to provoke the discussion, provoke the question. What does this mean? What does it mean to each of you representative, advocates, scientists, clinicians, people with SCI, biotech companies, device companies? What's the provocation here for us to address and dig our teeth into? That's what this was for. So I just want to reiterate that. We're going to take a break, and then we're going to come back. We're going to do that, classroom
Round robin.
exercise, where we can continue this conversation. So if you had questions, Trayvon, Susie, if you had questions that you wanted to ask, we're going to come back and continue this conversation amongst the others, or Jake and Jason and I'll explain when we come back. So get your break.
I don't
After the break.
I don't think you all will have to stay here during that part. I think
It's okay.